Welcome to this episode of PharmTech's Pharma Fundamentals. In this episode, Susan Schniepp, Distinguished Fellow with Nelson Labs and a member of PharmTech's Editorial Advisory Board, and Amnon Eylath, President and Founder of Broad Spectrum GXP, discuss biologics development and the Parenteral Drug Association's revision to Technical Report number 56, which Eylath is a co-author on.
My name is Sue Schniepp, and I'm a Distinguished Fellow with Nelson Labs. And joining me today on Pharma Fundamentals is Amnon Eylath, and he is the president and president and founder of Broad Spectrum GXP. And we're here today to study or to talk about the Parenteral Drug Association Technical Report number 56. I'm going to let Amnon go through the title and explain a little bit. I know this one was first published somewhere around 2007, and it has recently been revised. And so what's the full title of this technical report, Amnon?
Yes.
It's a long one.
Sure thing. Technical Report 56 is basically, it is application of phase-appropriate quality systems and GMP, and basically titled to focus on biological drug substance, but at the same time, it's been recognized that except for the biological part, everything else applies across small molecules and other areas. And to deal with things like cell and gene therapy, there's now a TR56-2 task force to focus on the complexities specifically for cell and gene therapy. But in general, it arose from my experiences at Eli Lilly way back, where the company was applying full commercial quality SOPs and standards to early phase clinical product, and it was not really working well. There are certain things that you just can't do or have in place. And I proposed a development quality system, which was accepted, and we worked on that internally and launched that, and that was adopted by the corporate quality. And then the decision was, if we're doing it, let's also get industry, FDA, EMA, other people on board, which I'm happy to say there was a lot of enthusiasm then. And we came out with the first one. We started working around 2006-07 and came out with the version formally in 2012. I updated in 2016 because of analytical method regulation changes in Europe. And we just finished another 2-year effort to really update it, current language, things that are now normal and commonplace, not have to be as emphasized, and also specific areas, a lot related to risk assessment and transfer of knowledge. So that's where we are now. It was just issued a few months ago, and we're happy to educate people on it, as we have, and we'll also be presenting about it with you moderating at the PDA FDA joint conference.
Yes, that's going to be an interesting discussion. In practical terms, there's not much, as you said, between a solid oral dosage form and a biologic in terms of how you apply the standard. So I guess I would say during the development phase of a biologic, it's a little more complicated, though the basic fundamentals of the TR apply to solid or small molecule. But there's that development of the biologics is a little more complicated. Can you go into where some of those complications are in the technical report?
Yes, absolutely. And I would say that the main difference, because I've worked extensively both in biologics and in small molecule dosage oral form, and applying these principles, is that the aspect of microbiology, sterility, containment is of a much higher level of rigor in biologics. So you have to have clean API, not contaminated, no toxic materials. You have to have clean formulated drug product, whether it's capsules or tablets, etc. But once certain things are proven, like that your process may destroy all bacteria or the product is too dry for antibacterial growth, you don't have to keep testing. And the containment aspect really has to do with cleanliness, obviously a clean workplace, no insects, rodents, dirt, or filth, as the FDA calls it, Particulates are of a concern, obviously, especially if they're metal or glass, but other things may not be as much of a concern, whereas biologics, even though your drug substance or API, depending on how it's called, active pharmaceutical ingredient, needs to be what is called low bioburden. Now, for all practical purposes, it is sterile, but there are regulatory and practical reasons to not call it sterile, because to declare something sterile needs validation and needs also extra very specific testing. But people, if they have contamination in whether it's a fermentation of bacteria or cells, eukaryotic cells, obviously you want to keep anything out of your cell culture. And in effect, you can't call it sterile because there are growing cells in it. So intrinsically, it can't be sterile. But we want low bioburden, no bacteria, no E. coli, no fungus, other things that could be dangerous to the patient and also destructive to your product.
What factors must developers take into account when developing a biologic so that the manufacturing process conducted down the line will, not experience delay or failures? But I also want to include in that what steps developers must take in terms of extractable leachables becomes a very different animal when you're talking about compatibility. I'm looking at compatibility of the glass and the vial and the stopper.
Yes.
So I know I threw a lot at you there,
That's
but
sure.
you can untangle it for
no
us.
problem. I mean, the general principles conceptually are the same. And I want to state that one reason I wanted this TR to be called quality systems, because quality systems does not just mean GMP quality systems. It also means quality systems like good documentation practice, developing risk management, transfer, archiving of information, which are all very important, and in many cases are often lacking from R&D. And the moment you have a beginning of a product, it's very important to manage it well, document it well. It just means quality does not have to approve everything or review everything. And the systems could be internal systems, business practices, or you may want to use some of your, like an electronic QMS if that's helpful, but it's not necessary. It's just what you have, a good system that will keep track of information. When you do your risk assessments, do them, document them, because one of the big pitfalls is when you get with any drug and you're getting from the research phase, maybe you're making some material for what's called toxicity testing, GLP testing for patient safety before you go into human, things may often be a disconnect. And the lack of transfer of information of both formal knowledge and tacit knowledge is really important. And that's why we emphasize it a lot in the new version. The manufacturing process related to biologicals is very different in a sense from the typical organic chemistry processes used for small molecules, in that it involves, in most cases, living cells, whether they're bacteria or eukaryotic cells, producing proteins and other materials. And the design of experiments around that is not as quickly done as it is in with organic chemistry, where you can do a lot of benchtop experiments quickly, and identifying, standardizing your medium, making sure that anything is not as variable, as low variability as you can, using defined media, very controlled conditions, helps in that. But it is definitely harder and takes longer to maybe optimize biological production. Now, when we're going from drug substance to drug product, and there's also aspects then, I suppose, with formulation in both small molecules and biologics, but the products are much more fragile, usually in biologics, and the conditions of storage and whether we're lyophilizing them or not, or freezing them, or they're very stable in a liquid form, as some biologics are. There's the whole aspect for sterile injectable, where we're talking about leachables and extractables, whether it's from a plastic syringe, or a glass syringe, or a glass vial, or a plastic vial. And a lot of that information can be obtained from the vendor. And the vendors who make these, just for example, Becton Dickinson makes a large amount of the world's, glass syringes, etc. Other companies making vials, stoppers, all of those have to be very carefully tested and proven to not contaminate or degrade, interact with the product. the vials and the stoppers themselves, or the plunger seal, if it's a syringe, have to be tested independently, specifically with the product. So you can get a lot of information, sometimes a large amount of information from the vendors. Some of that information may be confidential, but then you sign an agreement with them that they will allow FDA to review, let's say, their master file, their device master file for the syringe, for the stopper, for the plunger seal. But there is information that has to be done with the actual product. And that is because some products and their formulations could be acidic, could be a little basic. Many times there are surfactants, and they may actually extract materials from the plastic, from the vial, from the silicone rubber. So that is something that shouldn't be forgotten. I think it's always remembered, but I don't realize that people understand completely that the container closure system can, and in many cases can be, cause of a failure of batches or of the concept. And that's why that has to be tested earlier than you think, to make sure that what you have for containing your drug, biologic or small molecule, especially if it's in liquid form, is taken care of.
Let's go on to the regulations that govern biologics. We've seen some activity. FDA came out with a new guideline that's out for comment, or you can comment on it on their docket. There's also USP came out with some general information chapter on biologics. So, and I think the EU issued a statement. So the question I have is, what would you recommend are the most critical regulations and guidance that the regulators are offering, or the standard setting organizations that biological developers should be aware of?
Yes, I think the prioritization should be in which region one is working in. In the United States, then the priority is first to look at FDA regulations and guidance. What is good is that, at least for the past 10, 15 years, FDA and EMA have been aligning. We know that everybody is aligned also on ICH guidance. And some of these updates and changes, I think, are clarification of specific areas. I do think there are some things that came out with Annex 1 and came out now with additional requirements for sterile processes for isolators, that there is a lot of dispute, especially on the value of doing sterility validation for a isolator when it is open, when you know that once it is closed is going to be sterilized or sanitized using whatever means, whether it's chlorine dioxide or some other vapor, etc. And so there are lively discussions about that. I do think that what people are not happy about is that sometimes some of these regulatory guidances are not always very scientifically grounded. So they could be based on people's experience, maybe from whatever companies they've been working with, or if it's related to health authorities, what they've seen in inspections. But I do think, especially from the US, if any pushback goes against these regulations, it is that the desire for things to be grounded in science and not just in a compliance approach. But in general, I don't think all things are not necessarily barriers. It's more a matter that things are open for comment, and it's good that SMEs from around the world can opine into these new guidances and regulations. Now, if one is in Europe, obviously EMA, Udrelex, which are the equivalent of the CFR in the US, and everybody should align with the ICH guidance, whether it's related to sterility, related to, ICH Q7 is a great, important document, that one of the first that FDA adopted, simply calling it Q7, and many others. That you could definitely say that Q9, Q10 guidances related to assessing risks, related to quality systems, are universally accepted. So I would say, again, focus on your regional health authorities or regulatory agencies, and everyone should also focus on ICH, because that is truly universal and accepted.
Do you see anything coming out of PICS in the future that would kind of tie all the regulators together? I haven't seen that in particular. I don't know if you have any knowledge.
I would say that PIC-S, which is a global consortium of auditing, and things are coming out of that. are not official. Really, they are not regulatory guidance. They are best practices, and I think they're very important, especially in people referring to them, making sure that they are auditing things correctly and that they know how to prioritize when they're auditing or when they are assessing things. I find PICAS very helpful, but to me, it is not a regulatory standard that I follow. Same thing for World Health Organization. They cover a lot of things. A lot of the things are really, I would say, almost directly taken out of ICH and other type of guidance, but they are not legally binding. So it's more helpful. A lot of really good training materials out available from both PICAS and the World Health Organization. But the priority should be on FDA, EMA, and ICH.
Okay, let's move on to the quality systems piece because we haven't touched on that yet. What kind of, in a traditional quality system, what are some of the changes that a biological manufacturer might need to make to their to make it their quality system appropriate for biological development? Are there any big ones that jump out at you that aren't in a traditional small molecule QMS?
Not really. I think conceptually they're very much the same. It's more a matter that if you're putting a quality system, and to me, quality system is the governing documents, which are the policies, SOPs, work instructions. I also include things like specifications and forms into that. These are things that tell you what you need to do. Are very similar, very similar. What is different is obviously, as you mentioned, so the complexity can be greater in assessing a batch for a biologic, where you have to add all the sterility or low bioburden, containment issues, and also the fact that things may have to be processed a lot faster in biologics because sometimes you're collecting from a bioreactor to some chilled container, and you may have to go to the next step of production within 3 days, 7 days, etc. Usually in small molecules, you can crystallize things, etc., store them at -70, etc. That will not work for biologic intermediates. So I think that they are very similar, and that is why also the TR56, once you take out the sections there that are talking about cell banking and the importance of developing cell banks and tracking and all that, and things related to actual cell culture and low bioburden, very similar, if not identical, because they're conceptual.
A long time ago, I wrote an article for PharmTech that was on what's the difference between investigating a biologic and investigating a small molecule. And basically, my answer was the same as you. There really is no difference. You're going to follow the investigative process, but what you investigate is going to be a little different. And you're still going to drive to that root cause. What you said is you might have to do it faster because your go no go manufacturing decision is going to be on a different timeline than a traditional small molecule development. Am I understanding that
Yes,
correctly?
yes, and it's even more critical because certain intermediates or bulk biologics do not store well. And you may have to go from, let's say, your bulk drug biologic to lyophilization within 7 days and ship it over maybe to another country. And if you have an investigation and your product is stuck, then there can be a decision to forward process it at risk, which, by the way, is very expensive. But to losing a full batch of a drug substance, bulk, is also very expensive and would delay things. And some biologics are not always in plenty, meaning it's a complex process, and it may be only 1 or 2 batches away from shorting the market and the supply. So that's why there's a pressure that we can't just freeze everything down and deal with it after the long weekend. You may have to sit and immediately bring in the SMEs. And this is something that I was thinking as you're asking me the questions. One of the elements for success to me is that there needs to be a very good communication, collaboration between quality and the SMEs, or the people working in, whether it's manufacturing facilities, whether it's upstream, downstream manufacturing, drug product, fill finish, etc., there really needs to be good communication, collaboration. And I also find that it's very important that the people in the quality organization have an understanding and an interest and an affinity in the science and technology. Otherwise, it becomes a checklist operation, and it's not very productive. And I think that in my case, often as myself starting off as a biologist and always being a tech geek, interest in technology and whatever people are, how things work, is that I've been able to act as an interpreter or be bilingual and translate from the regulatory compliance aspects to people who are building or operating equipment, explaining why something has to be done, or then working with them to find an alternate way that is good, just as acceptable, would be acceptable to FDA or EMA, with the right documentation, the right experiments to prove it. And at the same time, also explain to quality that when you say you must sample from this biologic at a certain point in time, explain to them that is not feasible. We're not going to be opening the hatch on a product that is, for all essentials, sterile, no biologic, and risk that. Let's think out what other solution we have. How can we sample this? Do we have to sample at that point? Can we sample after it? And it's very important that the people in quality get that information, and then the same way. And it's really important to be able to have one foot in both domains of understanding the technology, whether it's, again, drug substance, drug products, sterile products, tableting, capsuling, because each one has their own challenges. People often think formulation for small molecules is easy breezy. It is not always the case. Some products, like some I've worked with, are stable as rocks. Other are not. And they have to be formulated in a very special way and maybe protected from moisture or oxygen. So those things that nothing is simple. And something I often tell people jokingly, I say, drug development is not rocket science. It's a lot harder than rocket science. You can't just do experiments and build things and prove things and rebuild them. It's very complicated. There are many still unknowns that could be impacted. The more you know about your product and the process and the environment, the better. But still things happen that are very odd, and you have to go to the X-ray cyclotron at the University of Edinburgh to do some high energy X-ray crystallography on your product to prove to the lab that, yes, this is not a contaminant. Same thing in the biologics. Sometimes you also have to make decisions based on investigations and inference, but it has to be very solidly grounded.
Thank you for joining us for PharmTech's Pharma Fundamentals video series. If you would like to suggest a topic for discussion or participate in an episode, please email us at ptprojects.com and include Pharma Fundamentals in the subject line. We look forward to you joining us next time.